Bismillah Rahman Rahim. Welcome again, guys. Welcome back to another video lecture by Kami Microbiologist. Today in this video we are going to talk about spliceosome mediated splicing. In the previous video lecture, if you guys remember, we talked about the RNA splicing overview, and we talked what is RNA splicing and how many types of RNA splicing we have. And before that video lecture, we talked about the post-transcriptional modification, and we talked about the 5' capping and 3' polydenylation in detail. If you missed this video lecture, it is present into my channel. Go back and watch it. Okay. So now, what is spliceosome mediated splicing and how it will occur, and what are the main requirements in this types of the splicing? We'll be talking all in this video lecture. So please stay tuned and keep watching. Okay, so let me write. Now, what is spliceosome mediated splicing? Spliceosome, first of all, we need to understand about the spliceosome. Spliceosome is nothing but it is the combination of 150 protein plus we have the small nuclear RNA proteins. Okay, now when these two complexes are bound together, then it will make a spliceosomes, and when among them there is also intron portion as well. So all these three things can combine together, will call that spliceosome. So what we have three main things, which is the intron, portion of the RNA, plus we have the small nuclear RNA protein, and we have the 150 other proteins. Okay, so it will make a spliceosome like structure. Okay, due to this spliceosomes, the splicing will occur. So let me once again tell you, it is the process in which the intron portion, which is the non-coding region which don't take a part in the protein synthesis, should be removed with the help of this complex, and the exon portion, which are responsible, which take a part in the protein synthesis, are connected to each other. So we call that spliceosome mediated splicing. Now before that, we need to understand some of the important terms, and we also want to talk about the requirements, that what actually we require in this spliceosome mediated splicing, and after that then we will talk about the mechanism. Okay, what happened if we talked about the requirements? So let me write. Requirements. So in this case, we require the spliceosome complex. Okay, among the spliceosome complex, what actually we have? We have 150 proteins, and we have SnRNA. Okay, now what are the SnRNA? SnRNA are the small nuclear proteins, small nuclear RNA sequences, which have almost 100 to 300 nucleotide genome. So these are actually the, if we talked about these things, so it is nothing but 100 to 300 RNA nucleotide proteins, RNA nucleotide genome sequences, and it can be denoted. And there are 5 SnRNA, okay? 5 SnRNA. So let me write 5. And it can be denoted like U1, U2, U4, U5, and U6. So these are the SNR, okay? SNRP. So it will make a complex which we call that SNRP. So let me write, it will make a complex which we call that SNRP. This SNRP stands for small nuclear ribonuclear protein. Okay, so in SnRNP, we have the small nuclear RNA sequences, as you can see, which is 100 to 300 long genome, and we have 150 protein. And this all things, plus in this case we have also the intron portion as well. Intron portion as well. So the collection of these things, the collection of this complex is called as spliceosomes. Okay. Now these are the main things that we required in the spliceosome mediated splicing process. Then we need to understand some important and basic terms here. So for that purpose, let me draw the RNAs, immature RNA or premature RNA, and in premature RNA we need to understand some basic terms due to which we will easily get idea about the mechanism of the spliceosome mediated splicing. So what actually we have the RNA, premature mRNA. So let me draw. Okay. As you can see, these blue rectangles are actually represents the exon. Exon. And this is the 5' to 3' RNA, and this is called as premature mRNA or pre-mRNA. Okay. This is called premature mRNA, and this is the 5' area, this is the 3' area, and these are actually exon. Now this one area we call a 5' exon, and this one area we call that 3' exon. Between them, this green one represents the intron. Intron. And all of you better know exon are the coding region which take a part in protein synthesis, and intron are the non-coding region which don't take a part in protein synthesis. So it should be removed or cleaved out. You know, between this exon and intron, there are a specific sequences which we call it GU, and this GU is actually called as 5' splicing site. So let me write. So between them we call it 5' splicing site, which is so between them we have the GU region. Okay. Now remember, with this 3' exon and between this intron or this 5' intron, what we have, this is the 3' 5' splice. So now remember, between this 3' exon and this intron, we have another splicing site. We call that 3' splicing site, and here we have the nucleotide, and we call that AG. So let me write. So at that specific position, as you can see, at that area, we have the 3' splicing site, splicing sites, and here we have the nucleotide like adenine and guanine. Okay. Then after that, we have another specific point in the intron, in the mid portion of the intron, or except the mid portion or inside the mid portion, we have another specific area. Let me write here. So here at a specific area we have the adenine molecule present, and we call that branch site. So let me write. These specific sites are called as branch site. Okay, remember guys, here at that specific area, this is the 5', so this will be the 3' area, and here we have the hydroxyl group. Okay, so this is the 5' area, this is the 3' area, so here we have the phosphate molecule, and with this adenine we have also the hydroxyl group present as well. So these are the main important terms that we need to understand before the spliceosome mediated splicing mechanisms. Okay, one thing that I forget about the inside the requirement, we have another things that we required. We call it branch binding protein. So let me write. We also required the branch binding protein, and we also required in this case U2AF protein or factor. A stand for auxiliary and F stand for factor. So we need these two things as well. Now what are the function of these things? We'll be talking later. So these are the two other requirements that we need in the spliceosome mediated splicing. Okay, so remember another important things here, that almost inside the this premature RNA, almost we have 97% are the intron portion, or we can say non-coding portion, and 3% are the exon portion, or we can say coding region, which take a part in the protein synthesis. So 90s, and among these three portion we have almost 2.5% region are just code for the protein. So just think about that. Okay, so this is the main things that we need to know about that. Okay, so now let's talk about the function of each protein, and then we will talk the mechanism. Now U1, the main function of the U1 protein, they will just recognize the 5' splicing site. So they will recognize that area. Now why they recognize that area? Because at that specific area we have GU region. So with U1, as it is the RNA, so definitely with inside this U1 RNA sequence is what we have the CA region. So if we have the CA nucleotide, so definitely it will be complementary and it will bind that specific area. So that's why U1 will recognize this 5' splicing site. Fine. Then U2 will recognize this branch site. So this U2 will recognize this branch site. Then second we have U4. U4 will inhibit the binding of U6. Now then we have U5. U5 is mainly responsible to bring 5' exon towards the 3' exon and connect it with each other. Then we have U6. What are the main function of U6? U6 is mainly responsible to form a laurent-like structure. Means laurent will be look like in this. So it will make a laurent-like structure in this form. So these are the main function of each SnRNA proteins or genome sequences. Okay, now then what are the mechanism, how the whole process or how the splicing spliceosome mediated splicing will occur? Let's talk about that. First of all, what actually we have this pre-mature RNA, and for the first protein that is activated, we call that U1. U1 will get activated and they will bind to that 5' splicing site first. Second, we have U2AF. It will get activated and bind to this 3' splicing site. And after that, another protein will get activated, which we call it branch binding protein. It will get activated and it will bind to that specific area. So we have three proteins come together and bind at that specific area. Okay, so let me draw each of the things that what happen. So at that specific area we have U1 bind here, and at that specific area we have U2AF, and then with this branch site, as you can see, so here we have the branch binding protein bind here. So branch binding protein will bind here, BBP. This is the first step. Second step is that after this recruitments, then we have some other, these U or proteins are get activated and bind here. Okay. Then after that, U2 will get activated and it will bind to this branch site. So it will replace the BBP. And once it replaces the BBP, so BBP will be released out and this one place is occupied by U2. Why? Due to this place, once occupied by U2, so then it will make a bulge-like structure like that. and after this bulge like structure, the U2AF is also get released out. So then what kind of structure we will get? We will get like that. So this BBP is released out, okay, and then here U2 will bind here, and it will make a bulge like structure, as you can see. So let's suppose this is the U2 protein, and due to this bulging like structure of U2 protein, which is made by U2, then it will also released out this U2AF. So U2AF is also released out. Okay, so finally we have just these two things, as you can see, U1. So what we have just U1 and U2, which make the bulge like structure, and due to this way U2AF is also released out. Second step is that. Third step what happened after that? Another three complexes or three protein complexes are get activated, and these are the U4, U5 and U6. U4, U5 and U6 are actually in a complex form, in a tri-complex form, as you can see. This is the U4, U5 and U6. So it is in a complex form. Why? Because as these are the RNA's genome, so definitely there are the some sequences which is complementary to U5, so it will make a complementary structure. There are the some sequences which is complementary to U6, so that's why it will make a complementary with this U6. And after that we will get a tri-complex, tri-SnRNA complex like that, and it will then bind to that specific area. So then after that it will bind to that specific area. So what actually happened? First of all, during this stage, the U1 will get released. Okay, so when the U1 is get released, so this place of the U1 will occupied by the U6. After that, with this U6, U5 is already bounded here, and then U4 will already binded. So what happened after that? U6, now let me write here, then you will easily get the idea about that. Okay, so this is the structure. So in this case we have U1 is already released out, so the U1 area will be occupied by U6. U6 are bounded here. With this U6, U5 is already bounded, as you can see. Okay, and here U2 is already present, as you can see. So this is U2, and this is the adenine residues or adenine nucleotide present. With this we have also the another complex U4. So this complex is formed, as you can see. Now what happened during this stage? The U6 will attack to this U2 or will attack to this A residues, because with this U6 we have the hydroxyl molecule, which is the hydrophilic molecule, and it will attack, so it will attack to this A. So with this A we have also, or we can say once this U6 will shift it to this A protein, so then a larynx structure will form, and due to this larynx structure what actually happened, this U4 is also released out. So what happened, the U4 is also released out, and we will get the structure like that. So how this structure will be formed? This structure will be formed that U6 will just shift it to that specific area with this UA. So let's suppose if this is the U6, so this will be the UA. Sorry, this will be the U2 having the adenine residues. So how these two things are come closer to each other? Because with adenine there is the hydroxyl group, and with this U6 there is the phosphate. So both are nucleotide and it will be connected with each other, because this U2 or this U2 have, or this U2 having the adenine residues, they have hydroxyl group, and this hydroxyl group will have a lone pair electron. This lone pair electron will attack to this alpha phosphate, or alpha phosphate, mainly it will attack alpha phosphate which is present to this U6. Due to this way, a Laurent structure is formed, as you can see. Now once Laurent structure is formed, so here we have also the 3' end, as you can see, 3' hydroxyl end. So they have also hydrophilic, and with this area we have also the, as I already told you, that U5 will always band with U6. So here is the hydroxyl group, it will attack to this 5' phosphate. So there is the 5' phosphate group present. So once it attack, this non-pair attack to this alpha phosphate of this first electron, so then what actually happen, it will be bring closer to each other. So it is possible by this U5 protein. So after that we will get the structure like that. This is the first exon which come closer. So there is the hydroxyl group and it will become closer to this another exon which have the phosphate. Okay, so both are come closer to each other and we will get the structure like that. So this is the mature mRNA. So in mature mRNA we have exons, as you can see, and this complex will be released out, as you can see. So it will be released out, and this is called spliceosome. Among them what we have the So among them we have U5, U6 and U2, and here we have also adenine as well, as you can see. So this is called spliceosome, so it will be released out, and this spliceosomes once formed and it will get released out. So what actually happened, it should be degraded inside the nucleus. So we don't need it, we just need this structure for the connection of exon portion with each other. So once the exon portion are connected to each other, so we don't need it further. So what happened, inside the nucleus this structure will be degraded, as they have 150 protein as well as intron portion and some other RNA sequences. All these RNA sequences are worthless, so it should be degraded inside the nucleus. After that, this mature mRNA leave the nucleus. Now let's suppose it is leave the nucleus and transfer to into the cytoplasm. So let's suppose this is a cytoplasm area. So inside the cytoplasm area, then it will give rise to protein. After that we will get the protein. So this is all about the spliceosome mediated splicing process. Hopefully you will get idea about that, but if still you have any kind of question in your mind, you can write it into a comment. I will try my best to give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and definitely subscribe the channel, and stay tuned, because in the next video lecture we are going to talk about self splicing in a detail. Thank you so much for watching.